chromosome is normally suppressed anyways. So we really only have one X chromosome that's supplying most of the genetic information that we need. So therefore, even if you're born with only just one X chromosome, you're not too greatly impacted. So that's the only reason why you sort of touch on that.
Oh, awesome. Thank you so much.
That's all right.
Do we want to go through the week 3 PowerPoint and then the Kahoot? Probably, right? Yeah, all right, sweet.
Well, I'll ask them. Do they just want to jump straight into the Kahoot?
Oh wait, maybe we can do the Kahoot and then go back to like week 3 if anything's like unclear.
Like we did last time?
Yeah. Wait. Oh wait, no, I'm on week 4. Give me just one sec. Oh, was it just genetics that we didn't cover?
Yeah, just genetics, like population,
yeah.
like Hardy Weinberg.
Yeah.
Yeah, and then chromosomal.
Oh, and then the HKS. Honestly, like guys, just read the slides. Like, it's fine.
All right, let's get started fairly soon, but not yet.
Is RJ, like, where is RJ? Is he celebrating his freedom?
I think so. I think so. But apparently, did you see Ryan was saying, like, as soon as he finishes quarantine, like the state, like Singapore's lockdown is going to start. So like, what's the point of him finishing it?
Oh well. He's enjoying his very short-lived freedom.
Yeah, I don't blame him.
Yeah, let him go out.
Let him live. Can someone else record as well? Like Chating or Heine or David, please.
Please not me, because I can't survive if it doesn't work again. Like,
Oh, no,
I feel like
recording.
Chating's...
I'm recording already,
Oh,
just
okay.
like in case.
I don't have enough storage left on this computer.
I can't just record on my iPad.
Okay, I can record. I'll record on this computer. Okay, my laptop might like run out of charge halfway through, but like whatever. Okay, it's recording.
Okay. I reckon we should make a start. You reckon?
Sure.
Yeah. Can you guys let us know in the chat if you want to go through the week 3 Kahoot first, or if you want us to finish off week 3 slides first? Just chuck it down in the chat real quick, and then we'll get started.
Guys, we need more
Need more than
opinions than 3.
Slides. Yeah, sorry. Yeah, we're doing
Okay, we'll do slides.
slides. Okay, democracy says slides. Okay, let's do this.
Oh, well, there's a lot of slides on genetics.
Yeah, there
just
are.
realized.
Yep, there are a lot. Do you want to start off with HKS, or do you want to? Yes.
I'll go through it very briefly because I don't think HKS is in the Kahoot, so it's like, how have your like HKS like shoots and like your lectures how have they been? What's like the general feeling people are getting? Chill? Okay. My 3 is a god, so like, okay. Oh, wait, let me keep the chat open. Fun times, okay. HKS is a pretty, like, it's, you know, it's an all right subject. I liked HKS. If we had Zoe here, Zoe would have a different opinion, but like, you know, it's not for everyone, but it is something that, especially in like times like now, and you'll learn in semester 2 as well when you go to population health, it is quite important. So we'll just go through it very briefly. A lot of the information, just go back and look at the slides again. So today we're just going to go over health determinants. So basically health determinants are, wait, sorry, let me move this. How do I move this?
We're into presentation mode, Heini. We'll deal with the chat.
Oh, yeah. Okay, easy. Okay. So basically, health determinants are factors that raise or lower the health of the population or an individual. So what we are mainly want you guys to focus on is this idea of like a biosocial framework. So it's the idea that, you know, it's not only just like biological factors that are going to impact someone's health. It's, you know, a lot of different interactions, both between someone's biological health, their social determinants, their socioeconomic situation. All of these different interactions within a particular system are going to impact people, and they're going to impact health systems, and then obviously from that they're going to impact individuals. So here's just a list. A lot of this has been taken from like my three's like slides and his lectures, so hopefully like you guys kind of know like what's going on, and you just need to be aware of what different factors are actually involved within health systems and how they actually interact. So we've got different determinants of health. Like I mentioned before, where mainly what they want you to kind of focus on, that's really like the important key things, is social. So obviously like we'll go through them, like biological causes, kind of obvious, just kind of within your body, within like an individual's health. Behavioral, also like kind of obvious. Social, you can think of them as like the causes of the causes. So basically it's the idea that different like health disparities, health inequality actually correlates to socioeconomic inequalities within groups of people. And this is all to do with like the biosocial framework, the biosocial theory. So social factors are as significant as biological factors. And then your last determinant is the actual physical environment. Okay, so with, I think David went through this briefly last time. I'll just like go through it again. Basically, you need to know these definitions, just know what they mean and know how to actually just talk about them. You don't need to, you know, it's not like, because I'm not sure how like your exams are going to work, and I'm not sure like how your assignments going to work, because obviously everything's changing a little bit, but just like have an idea what these kind of words mean, what these theories are, kind of the idea behind them. Don't kind of get too stressed, like too like worked up about it though. Okay, positivism. Positivism basically means that whatever is true can be verified through scientific experiment. Everything else doesn't exist. It's kind of hard to understand, but it's like if I said the sky is feeling sad today, like it can't be verified through like an actual scientific experiment. You can't actually like verify whether it's true or not. Therefore, it's non-existent, like it doesn't exist, that kind of thing. Inductive fallacy, basically, you would have seen in like Mythru's thing, it's like the swan. Like you would have seen like his black swan picture. So basically it's making a really broad generalization on like one specific observation. So like what Mythru's example was, all of the swans I have ever seen in my life are black. Therefore, like all swans have to be black, but it's like, it's not true. So that's like making broad generalizations. And monogenesis, it's an actual theory. So if you like search it up on Google, it'll tell you that it's the theory that all humans are descended from a single pair of ancestors. In like an HKS context, it basically believes that there's only like one single origin or like one single problem for a particular health outcome. So structural violence. So structural violence fits within this biosocial political framework. So structure just basically means it's like a local, national, international political framework. So, you know, basically like government kind of, you know, structure. And violence just means that it, you know, it harms people either directly or indirectly. So it basically just shows that structural violence is just talking about how, you know, if you've got like asymmetrical structures of power, how that can actually interact with different social structures and how that actually leads to health inequities and health inequality within individuals. Okay. This is taken directly from My3's slides. I just thought it was a pretty good example of basically how there's all of these different factors, all of these different things that actually lead to health inequity within health systems. So this is actually really like a really good kind of diagram. You'll find that My3, like his slides, are like really aesthetic, like they're really like on point. So if you ever see like him use like these diagrams and like flowcharts and stuff, like keep them in mind because they're actually pretty useful. Okay, so systems thinking, it's basically just you need to understand that within a complex system, which is basically like society, within society, connections, they're always changing, they're connecting, disconnecting. Everything is constantly changing, everything's constantly moving around. And they show this thing called ontological multiplicity. Basically, ontological multiplicity is that a single factor isn't isolated. Like you can't have like one thing that exists on itself. They exist as a multiplicity of factors. So here is an example. So like, for example, unemployment and social exclusion, they don't exist in isolation. They actually constantly are like intertwined. They constantly affect each other and they have, effects on health systems as a result. Okay, moving on. Just feel free to just like come back and look at these slides if like I'm going too fast. So then we move on to marginalization. So there's these two big fancy words. First one, hegemonic discourse, which is basically just like the majority, like the words of the majority, what people in the majority group are saying. And then there's marginal discourse, which is what the minority group is saying. I think I have more. Yeah. Okay, this took me a while to understand both like last year, and when I was revising these slides, I like kind of forgot like what it meant. Like it takes like a while to get your head around it. So there's this thing called epistemic injustice, which is basically injustice within systems and like thought structures. So you've got two different kinds. You've got testimonial injustice and you've got hermeneutic injustice. I don't know if I'm saying that right, but so testimonial injustice is basically unfairness relating to not trusting someone's word. So basically it's just if like a majority group just dismisses the minority group's kind of experience or like anything that they say purely because that they're like part of the minority, like just because they're a minority group. So we've got an example here. So if like a white person, I don't know like what else to call them, like a white person dismisses like a minority African American an account of being racially vilified, like they kind of just, like just not trusting someone purely because they're part of a minority group. And then you've got hermeneutic injustice. And basically that's unfairness relating to kind of, it's like, for example, it's like not trusting someone has experienced something, like not believing that someone has experienced a particular thing purely because that they haven't experienced it themselves, or whether it's like not like part of that particular culture. Like, for example, here, a woman who has experienced sexual harassment in a culture where there's no like concept of sexual harassment. So both, you'll often find that both the victim or like the minority group themselves don't actually understand that they have experienced that particular thing because it's not within their culture. And, yeah. Oh, wait, there's someone. Okay. Thanks, David. Yeah, and this was just something that I found in like a paper while I was trying to figure out what the hell these two things meant. Basically, the whole reason why that it's like injustice is basically it has effects on like health systems as well, because we don't like consider the fact that there are these two things that will affect people's like health experiences and how they actually affect health of individuals. So it's all about like this is like another example of like a social determinant of health. And then the last part of like marginalization marginalization is the whole concept of like constructing the other. So like constructing a minority group. So it's all about like how minority groups are presented by majority groups. Often it'll be in the media. Yeah, so you guys can like read that yourself. But that one's like pretty self-explanatory. Okay, genetics.
Yeah, I'll take this one. Okay. So genetics, we'll go through this. I understand that you guys had a lecture this morning on genetics, but we'll still go through it in a good amount of detail because these ones tend to be quite high yield, as usual. Not muted, am I? Yeah, okay, good. So if we talk about genetic disorders as an overall thing, the one we typically focus on is something we call a monogenic disorder. And this is one that just results from a single defective gene, and one thing stuffs up and the disease occurs. Other cases, you might get one stuff up and that increases your susceptibility to a certain disease. But the main important bit to take from here is that it's a single defective gene that causes an error. Okay. However, we also have things that are called single nucleotide polymorphisms, or SNPs for short. And essentially what these are, it's essentially genetic variation between the population, and it has to occur in at least 1% of the population to be considered an SNP. So what it is, it's just a small little difference in our nucleotide sequences, just from one single nucleotide. So for example, instead of an A at a certain location, it might be a T. And if that change occurs in at least 1% of the population, it's an SNP. Since our DNA has non-coding and coding regions, some SNPs won't actually play a difference since they're non-coding. However, some of them will play a difference because they are in the coding regions. We found these things out through something we call the Human Genome Project. Pretty self-explanatory thing. It's essentially scientists took a sample of DNA from a bunch of people, sequenced the whole thing, and made it available to the public. There are also studies where they kind of take samples of DNA from different people. It's just a research study, essentially. Not so useful. Pharmacogenetics are somewhat relevant, however, to do with SNPs because these SNPs can change how we respond to certain medication. So if one person has a certain variation, they might respond in a different way to certain medication compared to another person or the rest of the population. So what this means is that we can actually tailor our drugs towards certain populations or tailor our treatment to certain individuals depending on their genes. So instead of having a treatment that's kind of subpar and sub-quality, we can have a better quality treatment. On the side. Okay. So these are examples that you don't actually need to memorize. However, they gave some examples within our lecture to do with warfarins, which is a blood thinner, as well as a beta-2 adrenergic receptor agonist, which is something you'll learn later down the track. But essentially, different people respond to different drugs differently, and therefore we can tailor our treatment towards those different people. However, this stuff they won't ask you about in the exam. It's just way too detailed, not too necessary. And this is what we've been talking about. So SNPs can either leave you more susceptible to a certain disease, or it can also change our treatment. So in an ideal world, what we would do is essentially test the genetics of everyone and figure out whether they're better suited to something or worse suited to something else, and tailor our treatment or tailor our preventative measures towards that person. Okay, so that's like probably the lower end of genetics. This stuff is more useful. And we're talking about population genetics, so looking at the entire population and figuring out what genotypes and what phenotypes are best, or, sorry, not best, but most common, and calculating the likelihood of that genotype or phenotype passing down among the offspring. So just a few definitions here. Those who did bio 1, 2, and potentially 3, 4 would have covered this in a small amount of detail. So trait is a characteristic that you see, height, eye color, things like that, skin color. Phenotype is the appearance of that organism. So they have green eyes, brown eyes. The gene is on our DNA. Locus is just another word for location of a gene. Allele is a version of a gene. Genotype is our genetic makeup, typically dictated by capital letters and lowercase letters. A homozygote is when we have 2 of the same letter or 2 of the same allele, so capital A, or lowercase a. And heterozygote is a mix, uppercase and lowercase. And going from our genotypes, we can calculate genotype frequency or phenotype frequency. And essentially, this is just a percentage. It is a percentage of people with a particular phenotype or genotype. Fairly self-explanatory. This is how you calculate it. It's just the number of people with that particular genotype over the total number of individuals, multiplied by 100, and that gives you the percentage frequency. This equation, applies to both genotype frequency and phenotype frequency. The most important part of genetics, however, is this equation here. So the Hardy-Weinberg equation, which also looks like your, I think, addition of 2 squares. So p squared plus 2pq plus q squared equals to 1. So this equation is something that is a bit, it's hypothetical, it's theoretical, and doesn't actually exist in real life. And the main reasons for that is because it assumes that we're dealing with a large randomly mating population, and there's no mutation, no migration, and no selection pressures at all. So it's a very ideal situation, and this simply does not occur in real life. However, it does help us to predict a few things if we did have these conditions. And it will all be in something we call an equilibrium, if you remember back to chemistry. So everything's kind of staying the same. So in an exam, this is typically the tested topic of choice. And they'd essentially give you the frequency of one allele, frequency of another allele, and you'd be asked to calculate the frequency of a certain genotype later down the track. And I believe we have an example coming up. A few things can affect this equation, and the things that affect the equation are the things that are assumed to be the same. So if we get a new gene variation, that will change our equilibrium point and everything will shift around. Any migration may increase a certain prevalence of an allele or decrease the prevalence. Positive and negative selection will do the same thing. Genetic drift will just cause a sudden change in a certain allele and therefore change the frequency. And non-random mating will essentially make some things more favorable than others, and thus change the allele frequency. So if it changes allele frequency, then we can't have equilibrium, and therefore it will disrupt this Hardy-Weinberg equation. Okay, so we ripped through that, and we'll get onto chromosome disorders now. This stuff, there's only a few things to remember. However, the stuff that they do get you to remember is quite important. So out of genetics, I would say the Hardy-Weinberg equation is most important, followed by these disorders. We just have a few names here for different parts of the chromosome. So the P is the short arm and the Q is the long arm. They would have given you a huge list of vocabulary to remember in your lecture. However, most of it's not really required. P and Q is the most important bit. And then they tend to sometimes shorten down words into centromere down into cen, deletion into del, and so forth. However, they're pretty self-explanatory. It's just a shortening of the word. This stuff is most important though, the nomenclature on how we shorten down names, shorten down genetic conditions, sorry. So the first thing we do with this structure is we state the total number of chromosomes they have. So the normal number is 46, So if you see anything other than 46, you're going to know that there's a disorder already. The second thing we state is the sex chromosomes. So this can help us determine if they're male or female, as well as any disorders there. And then lastly, the third section of our name is the most useful bit because this will straight up point out any abnormalities. So in this case, with this first example, when it says plus 21, means that we have an extra 21st chromosome. And so forth. If it has del bracket 5p, that means we have a deletion on chromosome 5 on the short arm. So if you remember a few key words and a few key terms that help to direct us to the location of a gene, you'll be able to interpret these pretty easily. One main difference to point out, however, with these disorders is between the aneuploidy and polyploidy. So aneuploidy is the addition or subtraction of one chromosome. So for example, Down's syndrome is an extra 21st chromosome, whereas polyploidy is if we had a whole set of extra chromosomes. So for example, an extra first chromosome, extra second one, extra third, and so forth. And that is typically abnormal and the fetus won't survive that. However, aneuploidy, there are some instances where it's viable. We also have some classifications of our chromosomes, and these are natural changes and natural structures, so these don't affect function in any way. It's just essentially to describe the location of the centromere along the chromosome. They're best understood really through these pictures. So metacentric, M for middle, the centromere is located in the center of the chromosome. Submetacentric is if you move the centromere a tiny bit up, and then acrocentric is if you didn't have these short arms and just had these small little outgrowths of chromosomes. This picture is just showing a karyotype. So you would have seen this before. It shows all the chromosomes, checking the number, checking the shape, checking the length, and all of that. And we can also see our metacentric chromosomes, submetacentric, and acrocentric. So metacentric tend to be larger than submetacentric, and then to acrocentric. Karyotypes, you don't really need to know how to prepare a karyotype. Essentially, they take your cell, they break it down a bunch, they destroy your cell membrane, destroy your nuclear membrane, spread it out onto a slide, and then manually arrange your chromosomes. The most important bit to recognize is that it occurs with cells in metaphase. And the reason for that is because they're the most condensed at that stage. In your karyotypes, you can also see something called G banding, and it's this black and white or gray and black and white mix of bands on a single chromosome. So what it tends to be is that AT-rich regions, or adenosine and thymine regions, tend to be a bit darker than guanine and cytosine-rich regions, and then light bands consist of mostly active genes, so the ones that have been actively transcribed and translated. These G banding patterns can be used to match up homologous chromosomes together. We also have something called ideograms, and that's essentially just a map of a chromosome. So it'll help you determine at what location a certain gene is along your chromosome. Okay, so we went through this bit, aneuploidy and polyploidy. Aneuploidy is an additional or missing chromosome, whereas polyploidy is an additional whole set, an additional whole set. Monosomy, trisomy, tetrasomy simply indicate how many chromosomes there are. Mono being 1, tri being 3, tetra being 4. However, the more you add on, or the more disorders you add on, the less likely they're going to be viable, and therefore the less you'll see them in real life.
There's two questions just about karyotypes.
Karyotypes. Let me see if I can see it. Quadratics flashbacks. Peter, that's not a question. Concha, do you need to memorize the steps of preparing a karyotype? I never saw a question. I never saw a practice question on any questions I did. The only step that you need to really know is that you take cells that are undergoing mitosis in metaphase, and that's the only thing. Do we only need to know the words in bold for exams? Bold for exams, on the making a karyotype slide. Like I said before, you don't really need to know any of the bolded words. These are just the main things that they're adding. However, it's not really that useful. Yeah, overall very, very low yield. Study it if you really want to at the very, very end. Only if you want to though. Are there any other questions or anything before I move on? I see not. Okay, sweet. Okay, so these are types of aneuploidies or how they kind of come up. I believe we went through this during mitosis or sometime last week, I believe. And we went through the consequences of having non-disjunction in meiosis 1 versus non-disjunction in meiosis 2. And essentially anything that occurs earlier will be more fatal and will have worse effects in the long run. Yeah. However, this is a really important part. This will be, I think you guys cover it in an ICL sometime, and it's to do with mosaicism. So this is where for some percentage of a person, their cells are normal. However, for another percentage of their body, their cells have a genetic disorder. And this occurs if you have non-disjunction occurring in mitosis, so in your normal somatic cell division. So this one will lead to a bit of a mixed effect in the individual. You might get some different appearances depending on the location in their body. However, the effects tend to be a lot less compared to compared to non-disjunction if it occurred in meiosis. This here is probably the most useful bit you'll get out of genetics, and it's knowing some autosomal aneuploidies or additional chromosomes. So Down syndrome is the one you hear about the most. It's trisomy 21. It occurs quite a lot. Occurs a lot. They generally have a pretty good quality of life. However, there are cognitive and behavioral and physical differences that exist. Edwards syndrome is trisomy 18. This one, since it's an earlier chromosome, you'll have greater effects. So only about 10% survive beyond the first year. And I don't believe it's a great quality of life. And I think that you still do get some cognitive, physical and behavioral changes. And same with Patel syndrome, trisomy 13. Once again, 10% only survive past the first year. So yeah. The main thing to take into consideration here as well is something that will also come up in your ICL, but as maternal age increases, the chances of an autosomal aneuploidy increases as well. So typically we kind of put the border around 35 years old is where the risks of having autosomal aneuploidy starts to really increase quite a lot. Beforehand, it's fairly low and it doesn't increase as much, so it's pretty okay. We also have some sex chromosomes aneuploidies, namely Klinefelter syndrome and Turner syndrome. So Klinefelter is essentially when we have 2 X chromosomes and a Y chromosome. So and this results in a few problems, namely one being infertile, and males will have, and individuals with this defect will be considered male, however they have feminine features. And that's essentially due to a bit of a hormone imbalance. So it's typically treated with testosterone. Why does an extra chromosome cause these problems? Yeah. yeah, overexpression, cells divide differently as well, so that can cause some issues as well. But yeah, those are, yeah, Enoch and Akaksha explained it pretty well. And then lastly, the last sex chromosome aneuploidy you'll have to remember is Turner syndrome. And this is where you have a singular X chromosome. So this is what we were talking about a bit earlier before we got started with the presentation. We had a question to do with X chromosome deactivation, I believe. And the main reason you have to know that is because of Turner syndrome, where we only have one X chromosome. And this one, it occurs in females, so it's a monosomy of the X chromosome, and essentially you get reduced development of sexual characteristics. Yeah. Okay. There are a few structural chromosome abnormalities as well. These tend to be quite nicely named as translocation, insertion, deletion, duplication, and inversions. So translocation is moving something, insertion is putting it in the middle of another chromosome, deletion is getting rid of something, duplication is doubling, and inversions are flipped around. So, what the heck is this? Okay, right. The main thing to take away from these defects is what a balanced versus an unbalanced rearrangement is. So a balanced rearrangement is essentially just moving the location of some genes, but overall it doesn't affect the individual's expression of those genes. So you might not even know that you have a balanced rearrangement. And essentially the reason for that is because there's no net loss or no net gain of any genetic material. However, it can run into some issues in reproduction. So it could result in a partial trisomy. Due to the offspring gaining an extra section of a certain chromosome. If you compare this with an unbalanced rearrangement, this is where you get a net loss or a net gain of genetic material. And because you have more than you need, you typically get more clinical effects. And examples of those would be deletions and duplications. Okay. You also have different types of translocations. Not the most useful bit. However, we'll go through them. So reciprocal bits are just swapping, so you're just sharing quite nicely. Insertional is when you get deletion on one side and then insertion on another side. So you simply moved it along. And then you get a Robertsonian one, where you get two acrocentric chromosomes kind of fusing together at the centromere in the center. Something that I'm aware is confusing, however, is the difference between an insertional translocation and simply just insertion or just deletion. We asked our tutors, or I asked my tutor, and they weren't sure either. However, it won't come up in an exam where they make you choose between the two. It's just important to know that they are very, very similar, if not the same, and just two people who have named them. Yes. Okay, so this is talking about balanced rearrangements and how that can result in a chromosomal abnormality down the track. So what happens is essentially we have, what happens is you get a partial trisomy or a partial monosomy. So if you have a look at, say this one down here, we can see that what would be normal is just 2 pink and 2 blue. However, what we have instead is 2 pink, about a quarter extra pink, and then like 1 3/4 of a blue. So you get partial trisomy of pink and partial monosomy of blue, and that can result in quite a few issues down the track. So the main thing to take is that balanced rearrangements are fine for the person who has it originally. However, for their offspring, the effects could run a lot greater, and you might get some more significant developmental defects and cognitive impairments, behavioral impairments, and physical impairments. So that finishes up week 3. Quite a bit of information, quite a bit of confusing information as well. Does anyone have any questions? Otherwise, we'll be getting started on our Kahoot. So if you don't have any questions, you can start bringing up the Kahoot website. Otherwise, chuck your question down in the chat and we'll get to them. No questions. Okay. I'll screen share the Kahoot. So open up your Kahoots. Like last week, we'll be going through the questions after each one. And yeah. Just hang on. I'm doing it so I don't blast your ears out. There's the code 3782863. Let me know if the volume is all right. Hopefully I'm not deafening you all.
We can't hear it.
Pardon?
We can't hear your music.
Can't hear it.
I think it's just you. Oh, there we go.
Oh, okay.
Oh yeah, there's a question about insertional translocation here.
I'm answering it in the chat.
Who's RJ's hair?
Hmm?
There's a
Hey,
one.
you got a haircut. Isn't it a good haircut?
Yeah, it's good.
I agree. Love it.
Thanks, guys.
It looks good, yeah.
The
You
first thing I
finished,
did after 14 days.
Yeah. Peter.
Don't memorize. Honestly, yeah.
I think that's actually how you pronounce it though.
Peter.
Yeah.
It's true. Probably. Alright. Alright, join quickly. We'll be making a start because we have
Week
actually
4.
a lot of content to go through.
Yeah, we should have a few more people.
Yeah.
Oh yeah, and if you're still confused about the whole balanced carrier thing, that should have been a chute, if I remember correctly.
Alright. I feel like I can get started soon.
Do you want to just start and then people can just join if they're, because some people might have like
Yeah, true.
tuned out.
If you haven't started, if you haven't joined yet, that's too bad. Alright.
Newt.
What's the chromosomal abnormality for Edwards syndrome?
Don't check the chat for the answer.
It's a good way of remembering it, just like by the age thing.
Mm. I
No,
never
that's
realized
a
that.
good way. That's a good
That's actually
way.
a really good way.
No, there's another way by age, but it's like it like perpetuates really bad stereotypes. So I won't share that.
Okay.
I know the one
Yeah.
you're talking about.
Yes, I know. A senior
Okay.
told me, and I just
We're not going to perpetuate that.
Okay, so this one was pretty good. Monosomy X is Turner syndrome, and then trisomy 13 is Patau syndrome. Yeah, trisomy 18 is Edwards. Okay, good job, smiley face. Question 2. Which of the following is true regarding the Hardy-Weinberg principle? All right, sweet. Good, so mating is completely random. If natural selection were to occur, that means that you'd be changing your allele frequency because some have a survival advantage, whereas some have a disadvantage. And populations must be very small. That can cause some problems because it's a small sample size, and that means your mating will be slightly off, and you don't really have a sustainable population. So yeah, Hardy-Weinberg has a few requirements, and one of those is mating is random. All right. Gnocchi, come back. Okay, so translocation. Bad question. Bad question. This question should be, what is not? Wait, no, sorry. No, this question is perfectly good. Which is an example of an unbalanced rearrangement? So that means you get a net gain or a net loss of genetic material. And the only one that occurs in is duplication because you're doubling a certain genetic section. Inversion is still balanced because you're simply just turning something upside down. Reciprocal translocation are just two people swapping things, two chromosomes swapping things, sorry. So you still have the same number of genes. And insertional translocations, you take one bit out, you stick it somewhere else, and you still have the same total amount of genetics. So all those, red, yellow, and blue are all balanced, whereas green is unbalanced. Oh, see you, Gnocchi. Horse. Which of the following is true? All right, sweet. So, if we start off with all these answers, because biochem is a bit confusing, but it can't be red because it occurs in the adrenal cortex. Slightly different, you can remember it with cortex starting with the C and cholesterol starting with the C. Blue is not quite correct because cholesterol is predominantly packed into LDL. That's your bad cholesterol. Glucagon entering the pathway for cholesterol synthesis are joining two different pathways together. Glucagon is to do with glucose mobilization, whereas cholesterol synthesis is a different process. And then, yeah, HMG-CoA reductase is the rate-limiting step for the synthesis of cholesterol. And you'll learn later down in the track, towards the end of the year, that this is an important target for some medication that we use to reduce cholesterol levels. Good. Yeah. All right, so we just went through this one. All right, nice. Yeah, LDL's mostly cholesterol. That's the bad one because it spreads cholesterol throughout your body, gives you some blockages in your arteries, results in a heart attack. It's bad. HDL is the good one. Chylomicrons transport fatty acids. VLDL is a bit of a strange one. But yeah, LDLs are mostly cholesterol. All right, lives on top. Moving on, which increases fatty acid synthesis? All right, sweet. So increasing fatty acid synthesis. So glucagon is to do with your glucose synthesis, mobilization, turning glycogen back into glucose. Epinephrine will increase your blood sugar because that's an instant source of energy. So fatty acid synthesis takes a bit longer, so it's not as favorable for adrenaline. And then if we have a high-fat diet, if we already have a ton of fats, it doesn't quite make sense to synthesize more fats. So therefore, if we have a low-fat diet, we'll increase our fatty acid synthesis to make up for that decreased amount of fat. So therefore, it's green. Liv is still on top with yellow dog 15. All right, false. The appearance is to do with your phenotype. Genotype is to do with your genes. So therefore, this question is false. Or this statement is false. All right, Liv's got a pretty comfortable lead. All right, sweet. So once again, this is another rec questions based on of aneuploidies. So trisomy 13 is Patau's, trisomy 18 is Edwards, and 21 is Downs. So Turner syndrome is your X chromosome deletion. So that's where you just have a single X chromosome. So it's a monosomy XO or X0. All right, so this is a bit of a mixed question. Once again, coming back to biochem. Biochem is a bit scary. However, let's go through the answers. So in beta oxidation, that is the conversion from fatty acids down. Beta oxidation is to turn fatty acids into acetyl-CoA. 3 carbons produce acetyl-CoA, which enters the citric acid cycle, is not quite correct. Acetyl-CoA is a 2-carbon molecule. So therefore, it doesn't make sense to have 3 carbons producing acetyl-CoA. It occurring in the cytoplasm isn't the location that it occurs in. I believe it's in the liver, I think. Someone correct me if I'm wrong. But yeah, it doesn't occur in the cytoplasm. So 1 NADH and 1 FADH2 is produced per breakdown. Ooh, okay. Okay, so ketone synthesis does not occur when beta oxidation is occurring. So the correct answer is false. So when we say false, it means that ketosynthesis occurs when beta oxidation is occurring. The reason for that is that certain parts of our body can use ketones and aren't able to use fatty acids. And essentially, that means that beta oxidation can't be the sole process that occurs. The prime example of this is your brain. So your brain can only use glucose and ketones if it's in a starving state. So therefore, it can't use fatty acids through beta oxidation. Otherwise, you'll die. So ketone synthesis does occur when beta oxidation is occurring. Therefore, this question is false. This statement is false. Solid. All right, so cholesterol enters cells. So yes, it occurs through receptor-mediated endocytosis. So that's where a ligand attaches to a receptor on the cell membrane, and endocytosis, or bulk transport, will bring it in. And essentially, it has to occur in this process because your cholesterol is packaged into your LDLs. It has to occur through endocytosis because LDLs are simply too large to cross the cell membrane through simple diffusion, active transport, or facilitated diffusion. All right, 12th question. Okay. Let me think about this one. RJ, did you write this question? RJ there? Oh no, gonna have to explain it. Okay.
Yeah, okay, sure.
Yeah, okay, RJ, explain it.
Give me a
Thank
minute. Give me a minute, hold on.
you. All good.
Okay. So omega-3 is actually meant for maintaining lipid bilayer strength, and omega-6 are actually just precursors for longer fatty acids. So omega-6 is used for your what do you call that, paracrine hormones, which you'll learn further in immunology. But you just need to know for now that omega-3 is for blood clotting as well as for lipid bilayer strength. So that's why it's false. Yeah.
All right. Solid. Oh, damn. I think you're like 100% so far. But yeah, G banding. Okay, sweet. Okay. I'm fairly sure it's the opposite for option blue. Yellow is incorrect because the patterns are the same for homologous chromosomes, and that's why we can use them to match up our homologous chromosomes. And then green's the opposite way to the solution. Okay. How are there 26 answers and I'm placed 28th? Good question. I have no clue. Okay. So reciprocal translocations are once again the translocation where two things kind of swap. So that's what the statement says. So it has to be true. Reciprocal is just swapping. Okay, so undoubtedly it's a bit of a confusing question, but if you think about it, the position of the centromere is located between the middle and the end. So if you imagine a piece of string, you mark out the middle bit, the end of it, and you chuck the centromere in the middle of those bits. So at the quarter way mark, that would be sub-metacentric. Metacentric is just if it's between the two ends. So therefore it's sub-metacentric. Can't be acrocentric because that would just be the centromere is located at the end. Okay. Weak and strong paternalism was something we covered in ethics last week. David went through a bit of it. So red is correct because in weak paternalism, it's essentially where the doctor makes the decision. However, they make the decision when the patient is unable to or lacks the mental capacity. Whereas strong is kind of like it's considered a bit it's considered unethical because it overrides patient autonomy. Yellow is just not correct is not to do with their physical strength, it's to do with their mental capacity. And then green, poor evidence behind the decision. Evidence doesn't play a factor in paternalism. The only important factor that we consider is whether or not the patient is able to decide with a good level of mental capacity. And that's the main consideration we make. Okay, sweet. So not an epistemic trap. Red, blue, and yellow are the epistemic traps we were talking about. The solution is deductive fallacy because it's actually meant to be inductive. Slight difference, but it's important to take note of that. Deductive fallacies don't actually exist. That's just something I made up. Sweet. This is simply a percentage that you need to remember. The requirement is that it has to be in 1% of the population for it to be considered an SMP. All right, second last question. Oh, sweet. Good. So this nomenclature up here, 47XY plus 13, indicates a male with Patau syndrome. Edwards syndrome is trisomy 18. Frank is just something I made up. All right, last question. Something rearrangements are generally harmless. Nice, good. So balanced rearrangements are generally harmless, and good to see that everyone got that. Sure, congratulations, JS. Okay, let's keep moving. Does anyone have any questions about the Kahoot? Drop them in the chat, otherwise we'll be making a start on, oh goodness, it's so loud. Otherwise we'll be making a start on week 4. So good job, Liv, Luck, and JS, and to everyone else as well. As usual, all the questions that we put on the Kahoot will also be in Word document form on the drive. And hopefully there are no mistakes this time, because I didn't do it at a stupid hour of the night.
David. Oh wait, no, he's already answered. Don't worry. Move on. We can move on. Oh, epistemic traps. Oh wait, okay, I can go through that just quickly. So, where is the slides? Epistemic traps, you just need to remember those few. Feel free to just go back to the slides. Basically, it's like all in the slides. Yeah, I'll just like quickly go through it again. So these are the three. So positivism, inductive fallacy, and monogenesis. Is there one in particular that like people don't really understand, or like is it just like all of it? Like I understand like it's kind of like difficult at the start, especially if we go to like the epistemic injustice, like those two definitions. Like it took me a while to like even wrap my head around it. But you do just need like a general idea and like how it fits within like an HKS context, rather than like you don't need to be like an expert in it. You just need to be able to talk about how it relates to like health systems and how it like relates to like health inequalities and health inequities. But yeah, so all of the information is on the slide. So positivism, inductive fallacy, monogenesis, and then you've got the epistemic injustice, which is the testimonial injustice and hermeneutic injustice. I'm not sure if you can see this, but like I've put like notes as well for epistemic injustice, especially because those two are like, yeah, those two are a little bit more difficult. But yeah, just know them in the context of HKS and you'll be okay. But yeah, any other questions before we move on to week 4? Okay, do we want to move on to week 4?
I'm muted. Biotech is Jatin's, I believe.
Yeah, are we going to do biotech today? Yes?
Yeah, I think that was
Okay.
the most confusing lecture. Yeah.
Okay.
Thanks.
Yep. Share screen. One sec, just let me put my cat to sleep.
Oh, that's so cute.
Look, I'm bored, it's quarantine. Browser pets. Okay, so biotechnology. For most people who have done Bio 3-4, you should have probably a better foundation of understanding this, but we'll go through everything. So the first thing that we're going to talk about is recombinant DNA. So first of all, what is recombinant DNA? All you have to know is that it's essentially you're creating one DNA, but it's essentially come from DNA from 2 different sources. So you're essentially combining 2 different DNA into one single vector. So how do you actually make recombinant DNA? So the first step is you have to have a vector. This can be a plasmid, which is essentially those little circle rings of DNA that you can find in bacteria. They can also be a virus, or it could be an artificial chromosome. Most of the time you'll deal with a plasmid. And in order to insert a foreign piece of DNA into your vector, you have to essentially cut your vector and cut the DNA you want to insert, so that they're both now open and you can stick them together. Now, the enzyme that you use to do that is called a restriction enzyme, and they cut at specific recognition sites. Now, restriction enzymes can cut to form 2 types of ends. One is called a blunt end and one is called a sticky end. So a blunt end is when it just ends up straight, and we don't really want blunt ends when we're making a recombinant DNA because these guys are hard to stick back together. So we want to form sticky ends, as you can see in the diagram. So it's kind of like a zigzag shape. Because there's a little bit of an overhang here, it's easier for us to then join back these 2 separate pieces of DNA together. So we want to form sticky ends. And then after we form these sticky ends, using our restriction enzyme, we then use another enzyme called ligase to then rejoin these DNA together, one from the vector and one from our desired DNA, and they join back via covalent bonds. So when we're selecting our vector, there are certain criteria that we can consider. So typically when we're looking at a vector and we choose a plasmid, we want there to be a replication unit or an origin of replication, so that we can, after we've made one vector with a recombinant DNA, we can essentially just let it go off and replicate itself. So then we can get thousands of recombinant DNA without having to go through those steps again. They should also have recognition sequences so that our restriction enzymes can actually act, because restriction enzymes aren't going to cut the DNA unless they find those specific recognition sites. We also want something called selectable markers, because these will help us later to identify whether or not we've actually been successful in making a recombinant DNA or not, because sometimes the process can fail. And one of the most common selectable markers that we use is an antibiotic resistant marker. So an antibiotic resistance marker is essentially just an antibiotic resistant gene, and these will code antibiotics that will be able to break down bacteria. So only cells that are successfully transformed by the vector, so in other words, they contain the recombinant DNA, only they can grow on a medium containing the antibiotic, because they now have this resistance gene inside of them. For example, this is a typical example that faculty likes to use. The ampicillin resistance gene AMP codes for an enzyme beta-lactamase, and this essentially destroys the antibiotic. And once again, the main purpose is to distinguish cells that have successfully undergone transformation compared to those who have not. Okay. So we use recombinant DNA in certain medical therapeutics, and one example that we like to use is tissue plasminogen activator. You don't actually have to know the exact example, but you do have to know how we go from making a recombinant DNA to essentially actually getting the protein we want in the end to treat our patient. So if we look at the steps here, these first few steps are exactly what I have been talking about up until step 3, which is where we've now included our foreign gene or our desired gene that codes for our TPA into our vector or our plasmid. The next step is we have to insert this plasmid into a cell, which we've chosen bacteria here. And then after we have successfully transformed it, then essentially we use bacteria as our slave, and it'll quickly produce a ton of the protein that we want. And it'll produce the protein that we want because it's got the foreign gene inserted into it, which it didn't originally have. And then we will isolate the protein, extract it from the bacteria, and then be able to give it to our patient after it's extracted. So the main thing is not what tissue plasminogen activator is. The main point here is how do you go from having a plasmid to inserting our desired gene to then transforming it into or putting it into an actual cell to then extracting the desired protein we now have made and giving it to patients. And also, once again, so which vector should be used? So when it comes to therapeutics, another criteria that you have to consider when choosing your vector is what type of protein that you want to produce in the end. So the choice of vector will largely depend on the properties of the protein, as well as particular features of each host cell system. So for example, bacteria and yeast are easier to grow than mammalian cells. However, mammalian cells handle expression of larger proteins better than bacteria or yeast. Mammalian or yeast cells are also more suitable for expression of proteins that have post-translational modifications. So if you're trying to produce a therapeutic and you know whatever protein you want to produce in the end requires post-translational modifications, you would need to choose either a mammalian or a yeast cell to put your plasmid in, even though bacteria typically are easiest to grow. And also mammalian cells are more expensive to use, but that's not the important part. I've put the important ones in bold. Okay, so next is farming. And this is essentially the process of genetically modifying plants and animals so that they can produce whatever we need to be used as pharmaceuticals. So for example, we could use sheep. So we start off with an expression vector. It carries the desired gene, and we essentially put that into an animal egg. Now we put the egg into a surrogate mother. Now the surrogate mother will have offspring, and these transgenic offspring produce the protein in their milk. Okay, so we're not, we don't get the desired product straight away from the mum. We have to wait till we have their offspring, and the protein will be created in the milk. We take the milk and we extract the protein we need, we purify it, and now we're able to give it to patients. So that's the process of farming. Another thing you guys need to know is polymerase chain reaction. They like to test you a lot on this. So the purpose of polymerase chain reaction, or PCR for short, is essentially want to rapidly increase or produce a large number of copies of a particular genetic sequence. So PCR has 3 main steps. The first step is denaturation. So the purpose is to denature our double DNA strands into single strands, and we heat it to 94 or 95 degrees in order to denature it. The second step is called annealing. So what we do here is we have to lower the temperature back down to around 55 degrees, and this will allow us to add these short primer strands. If we kept it at 94, these short primer strands wouldn't be able to be added on because the temperature is too high, and it'll just denature right back off into single strands. The last step is called extension. So we reheat it back to 72 degrees, and we add something called Taq polymerase to synthesize new DNA. So Taq polymerase is essentially just a special type of RNA polymerase that we've obtained from bacteria that essentially live in really hot environments. So it's already used to high heat, and so it won't denature at a temperature of 72 degrees, because if we used normal human RNA polymerase, we chucked it into 72 degrees, the enzyme would denature and it wouldn't work. So that's why we have to use Taq polymerase. This same process, steps 1, 2, and 3, is repeated over and over again, and the number of copies essentially increase exponentially. Yeah, so we start off with 1, we go to 2, we go to 4, 8, 16, etc. Okay, so a bit about cell differentiation. So as cells differentiate, the more differentiated they become, the less types of tissue they can produce, essentially. So you start off with totipotent cells. So these essentially retain the ability to give rise to all embryonic and extra embryonic cells and tissues. So they can pretty much do anything. And only cells that are the fertilized egg, the zygote, and early morulars are totipotent. Don't worry about this last one, just know fertilized egg and zygote. And then afterwards, they become pluripotent. They're still pretty good at this stage. These cells can form pretty much all somatic cells and germline cells of the developing embryo. So the only thing they can't form are extra embryonic tissues. So embryonic stem cells are pluripotent cells. And the next stage down is multipotent cells. So they can only give rise to cell types within their lineage. So most adult stem cells are multipotent cells. Okay, so therapeutic cloning. The purpose of therapeutic cloning is to derive embryonic cell lines with the same genome as the nuclear donor. And it's achieved essentially via 2 ways. The first way is called somatic cell nuclear transfer, or SCNT. And this is where we actually generate embryonic stem cells, so like the real thing. And then we induce them to form whatever specialized tissue we need it to differentiate into. Or we can do genetic reprogramming. So we essentially take an adult cell and we trigger certain genes so that it essentially goes back in time almost to form what's called an induced pluripotent stem cell. And then from there, we can then induce it to form different specialized tissues. Now, I just want to point out that therapeutic cloning is different from reproductive cloning. So the goal of reproductive cloning is to actually create a new organism, so a new person, a new dog, whatever. Whereas the goal of therapeutic cloning is we're only trying to create cell lines derived from an embryo. So we're not actually going to take it and transfer it in utero. So starting off with SCNT. So the process of SCNT is to make customized embryonic stem cells. So the first step is we have to remove the nucleus from a recipient egg. Then we extract the nucleus from a somatic cell, normally from the patient that we're trying to treat. This nucleus from the patient is then inserted into the recipient egg. So this egg now contains the patient's genetic material, not its original genetic material, because we've removed the nucleus in step 1. And then this egg is stimulated to divide, and it will form a cluster of cells known as the blastocyst, which is essentially just an early embryo. Now, the next few steps are taken to produce our customized embryonic stem cells. So our embryonic stem cells are isolated and they're collected from the blastocyst. And then they're cultured to create specific lines so we can induce them to form specialized cells. So if we needed, let's say, a liver cell, we'd induce it to create a liver cell. If we needed skin cells, we'll tell it to differentiate into skin cells. The advantages of this is we can treat disease of pretty much any body organ because these cells are pluripotent. And the goals of this essentially is we're hoping in the future we can reduce transplant wait times, and we also reduce the risk of immune rejection because remember, the nucleus we got was originally from the patient. So you don't have any risk of immune rejection, unlike if you were to get a transplant from another person. Now, the main disadvantage for SCNT is that there are ethical concerns related to the fact that we're essentially creating an embryo, we're letting it grow to the point of a blastocyst, and we're almost pretty much killing the embryo just so we can get embryonic stem cells to treat patients. Yeah, and so that was the steps outlined in diagrams. Yeah, and so because of the ethical issues, we've essentially created another way or another option that we can use, which is called induced pluripotent stem cells, or iPSC. So what this does is it takes differentiated cells, we alter the genes, so we essentially undifferentiate them to try and make them behave more like stem cells. Scientists essentially found that there are 4 genes that code for specific proteins that control these genetic switches. And so we use these 4 genes to our advantage to try and undifferentiate them back to behave more like stem cells. Now, the main challenges of iPSC is that it's not always successful. There are always going to be epigenetic differences. So although we've tampered with these 4 genes, they're not always going to reset to be completely like embryo cells. And there are also genomic abnormalities. So a lot of the times when we do tamper with genes, unfortunately, iPSCs typically incorporate extra cancer-causing genes, and there are also fewer tumor suppressor genes. So there is a higher risk of cancer, essentially, if you do transfer them back into the patient. And so this diagram here is the steps. So let's say we first remove the skin cells from a patient, we grow them, and then a vector that carries the genes, which controls how differentiated cells are, so we add them, and then those genes will essentially trigger them to undifferentiate back, and so now we have cells that are growing essentially as pluripotent-like cells. So now we've essentially come from non-stem cells to an iPSC. So gene therapy. So gene therapy is changing an allele from diseased to healthy. We require a recombinant virus to almost act like a vehicle to infect the patient. And the treatment is done via introducing a normal allele into the patient who would otherwise have defective alleles. Unfortunately, the main disadvantage for gene therapy is that it doesn't last long because we haven't fixed their original defective allele. We've just added in a temporary new allele that will produce proteins over a certain period of time, but unfortunately, the proteins do get degraded over time. So for example, here we have a sick patient who has a certain deficiency. We put the normal allele, we put it into a vector, so it could be a virus. And then the virus will essentially incorporate into one of the patient's cells. And because we're carrying the normal allele, the cell is able to read this allele and produce whatever protein that this patient is deficient with, and they are essentially well for a short period of time. So it's only there to relieve the symptoms, but it doesn't actually cure the cause. And it's still not widely used. In fact, I don't even know if it's used at all currently, because there are still several improvements that are needed. Another one is gene editing, which is done with a technology called CRISPR-Cas9. So it's essentially a recent method that we've discovered, which allows us to incorporate or transfer specific DNA. So instead of temporarily adding in a new allele like gene therapy, this allows us to actually go in and correct or replace the original defective allele in the patient. So it's almost like we can now solve the root of the problem. So the challenges that come with using CRISPR-Cas9 is that there is the potential that the edits could occur in the wrong place. So the enzymes that cut the DNA, the nucleases, could cut the wrong location. And there are effects such as it might cause cancer, or we might end up overproducing a certain protein that we don't need. Okay, and that was all for biotechnology.
Okay.
Who was doing Hunter syndrome?
I'll do Hunter's, yeah.
Yeah.
Okay, so Hunter's syndrome is quite low yield. So there's just a few things that you need to know. It's firstly that it's almost exclusively in males, so it's X-linked recessive. And the pathophysiology of it is basically it's a lysosomal storage disorder. So non-degraded material just accumulates in the lysosomes of these affected individuals, and then it builds up and it affects your body. So usually there's a mutation in your gene that encodes this enzyme. So this enzyme is meant to break down excess GAGs, and so when they build up, it just causes a lot of side effects. And you can see in the next slide, there's a lot of signs and symptoms. So usually these signs are not apparent prenatal or at birth, so it's quite hard to diagnose. But after a few months, and especially after the second year, there are unusual facial features such as a broad nose with flat nostrils, enlarged tongue. Your abdomen is swollen due to enlarged liver. And one main thing that is really noticeable is that individuals will have scoliosis or kyphosis. So scoliosis is just a curved back, and a kyphosis is hunched. And then this is because excess GAGs result in joint stiffness. And neurological problems that progress to mental impairment. For the attenuated form, there's no mental impairment. And another main thing is that individuals will also have difficulty walking, and they tend to walk on their toes due to the tightness on their Achilles tendon. Okay, but you don't need to know all of these things. You just need to know the main features to help recognize and diagnose an individual. That's basically it for Hunter syndrome.
Sweet. So it's getting, we just had a question in chat. What does X-linked recessive mean? It means that the defect will occur on your X chromosome, and recessive means that you need, you essentially need 100% of your X chromosomes to have that defect for the clinical presentation to show. So for males, that means just having an affected single X chromosome, and for females, both of their X chromosomes being affected. So it occurs more in males because we only need one X chromosome, whereas in females, it's a bit more difficult to occur. Attenuated form, RJ?
Hold on, let me see the chat.
So attenuated form, was it attenuated form of the vaccine? No.
No.
Attenuated, no?
It's the attenuated form of the disease. So you can have different levels of the disease. So if it gets progressively worse, you can have mental impairment, but some individuals may not suffer from mental impairment.
So attenuated just means it's still living, so it's still a live version, but it's just greatly reduced in terms of its severity.
Yeah.
Yeah.
Sweet. Okay. It's getting pretty late already, and we understand you guys have lectures early in the morning. If they're still live and if you rock up for 8 a.m.s. So I think we'll probably close it up there, unless you guys have any questions. But before you guys go, please don't go yet.
Why did you not tell me you were all going to wear it?
Wait, get your med hoodie.
Oh.
Sorry, that's not what we're gonna do. But we have a feedback form in our Google Drive, and I just sent it in the chat. So if you guys could fill that out, that would be like absolutely amazing. We'll also continue on with our week 4 content in our next session, and we'll update you with when that is. We'll upload our Kahoot questions onto the Google Drive. And as usual, if I've made a mistake again, text me or someone, and we'll fix that or explain the question. Does anyone else have anything to add?
We still have a Kahoot next week.
Yes, Kahoot next week as well, for week 4 content. So this will become a regular thing. Yeah, does anyone have any questions before we finish up? Otherwise we might do any plans for the break? That's a good question.
Catch up on all the lectures I should have watched since week 1. So
Yes, Heidi, same.
For the PSP. We are trying to figure it out. But we'll let you know.
Maybe we should do another.
David.
Okay, sure, David, sure.
Yeah, maybe. But we'll see. We'll let you guys know. But it should be a good time. Any other questions? Yes, under the wraps. It's so under the wraps. We haven't even talked about it yet.
Literally haven't.
We haven't even talked about it.
Yeah. All right. If there are no questions, we'll upload this recording onto the Google Drive probably tomorrow morning or something. So yeah, otherwise slides are up on the Google Drive, topical and weeks, and then yeah, fill in our Google form, please. It would help us a lot. And be honest. Thank you. All right.
Stay safe, guys. Make sure you stay warm.